today's webinar. I would like to seek your cooperation in completing this webinar. Kindly mute your mobile phones to avoid interruption. Thank you for your kind cooperative and attention. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. My name is Dr. Bunga Indah Kalkausari. It is a wonderful and precious chance for me to be your master ceremony this beautiful morning. First of all, let's say thanks to the Almighty Allah Subhanahu wa Ta'ala, who has been giving us guidance, happiness, healthy, and mercy, so we can attend and participate in this even without any obstacles. Praise and salutation upon our Prophet Muhammad Sallallahu Alaihi Wasallam, the best figure of this universe, the person who was able to save us from Jahiliyah era. In this beautiful morning, I would like to welcome Honorable Guest Lecturer Professor Dr. Christoph Herwig, full Professor Bioprocess Technology, Senior Advisor Corber Pharma Software. The Honorable Professor Dr. Chandra Yoga Aditama, Specialist Paru Konsulen, Mars, DTMNH, DTCE, FISR, as a Director of Pasca Sarjana YRC University. And respectable to Ms. Hilyatus Zahroh, MPKM, MSE, as our moderators today. Ladies and gentlemen, welcome to today's webinar Expert Lecture Magister Science Biomedicine. Today's webinar will discuss about introduction to digitalization for solution biomanufacturing of regenerative medicine. We would like to thank everyone here for making time to attend his remarkable webinar. Ladies and gentlemen, before we begin this event, let's reciting Basmalah together. May Allah Subhanahu wa Ta'ala make this event beneficial for all of us, and this event can run smoothly and conducively. Bismillahirrahmanirrahim. On this special morning, we have several agendas, so allow me to read several sequences of our agenda. The first is the opening, which we previously had read together. The second is reciting Holy Quran. The third is singing Indonesia Raya. The fourth is welcome speech from Director of Pasca Sarjana YRC University. The fifth is a core event webinar about introduction to digitalization solution for biomanufacturing of regenerative medicine. The sixth is a closing. Step on the following agenda is reciting the Holy Quran, who will be recited by our brother, Dr. Yufo Parigi. Doctor, time is yours.
Thank you, Dr. Bunga. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. Ya ayyuhalladzina amanu idza qila lakum tafassahu fil majalis إذا قيل لكم تفسحوا في المجالس فافسحوا يفسح الله لكم فافسحوا يفسح الله لكم وإذا قيل انشزوا فانشزوا وإذا قيل انشزوا فانشزوا يرفع الله الذين آمنوا منكم والذين أوتوا العلم درجات والله بما تعملون خبير. Aku berlindung kepada Allah dari godaan syaitan yang terkutuk dengan menyebut nama Allah yang Maha Pengasih dan Maha Penyayang. Wahai orang-orang yang beriman, apabila dikatakan kepadamu, berilah kelapangan dalam majelis-majelis, maka lapangkanlah. Niscaya Allah akan memberi kelapangan untukmu. Dan apabila dikatakan berdirilah kamu, maka berdirilah. Niscaya Allah akan mengangkat derajat orang-orang yang beriman di antara kamu dan orang-orang yang diberi ilmu beberapa derajat. Dan Allah maha teliti apa yang kamu kerjakan. Maha benar Allah dengan segala firman-Nya. Sadaqallahul azim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thanks to our brother, Dr. Ufo Parigi, who has recited the Holy Quran for us. May Allah always give us guidance and help. Amin. The third agenda is Sing Indonesia Raya. Ladies and gentlemen, the fourth agenda is a welcome speech from Profesor Dr. Chandra Yoga Aditama, Spesialis Paru Konsulen, Mars DTMNH, DTCE, FISR, as a Director of Pasca Sarjana IRC University. For Mr. Professor, time is yours.
Thank you. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi, good morning, goedemorgen. Let me begin with a speech in bahasa. Jadi tentu pertama-tama kita panjatkan puji syukur kehadirat Allah Subhanahu wa taala, Tuhan Yang Maha Esa, karena kita dapat berkumpul hari ini untuk webinar tentang dari program studi biomedis ini. Seperti teman-teman ketahui, pada saat yang sama sebenarnya juga ada rapat MOR ya di IRC, tapi saya sudah minta izin karena saya kebetulan ada dua acara, selain ada acara ini ada acara rapat ASEAN. Saya hanya ingin sampaikan tiga penghargaan begitu. Tentu pertama penghargaan kepada teman-teman saya di program studi Magister Biomedis yang sudah menyelenggarakan acara pada hari ini. Saya yakin ini acara yang sangat penting untuk kita menimba ilmu, apalagi kebetulan ada tamu yang bisa diajak untuk share ilmunya pada kita. Yang kedua, terima kasih penghargaan kepada peserta. Saya lihat sudah lebih dari 40, mudah-mudahan nanti terus bertambah. Saya yakin kita dapat menambah ilmu, menimba ilmu dalam acara webinar pada pagi hari ini. So, as I mentioned earlier, I just would like to appreciate the organizing committee for organizing this webinar, and also appreciating the audience of this webinar. Also, I would like to thank and appreciate Professor Herwig, who will share with us a guest lecture on this topic. I just read the title of the guest lecture, Introduction to Digitalization Solution to Biomanufacturing of Regenerative Medicine. I just take three kind of keywords from this title. The first one is regenerative medicine. Second one is biomanufacturing. And the third one is digitalization. These three words is important, of course, for us in the Universitas CRC. In our biomedical program, we have magister as well as the doctoral program of biomedical, as well as we have faculty of medicine. So I'm very sure that your lecture will be very, very valuable to us to enrich our knowledge and also in this topic. But I would like also to share with you, Professor Herwig, and also to all of the participants, the topic, at least the digitalization, biomanufacturing, and regenerative medicine are in line with the Ministry of Health's national program. Now the Ministry of Health, they have they call it six health transformation. And transformation number 6 is about health technology. So the use of digitalization and advanced technology and biomedical engineering, those kind are all in line with the health transformation number 6 of the Ministry of Health. So once again, thank you very much. Thank you well, Professor Herwig. So I'm sure your lecture will be valuable not only for our campus, our Universitas CRC, but also from the health of Indonesia. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Professor. The next agenda is remarks. Ladies and gentlemen, I immediately invite the moderator to host this webinar. Please welcome Ms. Hilyatul Zahra, MPKM MSc. Time is yours.
Thank you very much, Dr. Indah. Hi, everyone. I'm the moderator of this talk. Welcome to the webinar, Introduction to Digitalization Solution for Biomanufacturing of Regenerative Medicine. Our speaker today is Professor Christoph Herwig. He's a full professor for bioprocess engineering at Vienna University of Technology, and he is also senior scientific advisor in Corber Pharma. He has history of working on the link of science-based development to scalable manufacturing of bioprocess. And he really has strong engineering professionals skilled in research and development, life sciences, bioprocessing, validation, and GMP. So without further ado, let's give Professor Christoph Herwig welcome.
Thank you so much for inviting me, having the possibility to talk to you, to meet you here, also to e-meet you. It's a very big pleasure to me. Thank you so much for organizing this. As we have a hybrid solution here, I will keep seated and try to explain what I want to show you here with sharing my screen. I hope you can see my screen. You can see me. Yeah, thank you so much for being here. So what I'm going to do here is very much related to what I have experienced in my career. So I have been very much in biopharmaceutical industry, and if you have a bottle, and this bottle is full of protein, of purified protein, how much would that be? What do you think? Will be probably $2 million, something like this. And myself, I have put that away in the drain because the quality was not okay. So what to do next? What was the question? Was not why did it happen or what did go wrong? My colleagues in industry mainly asked me, when do we do it again? and let's hope it will be better. And that's not the way to work, okay? That's the reason why I actually changed to be professor and leave industry and finding methodologies to improve biomanufacturing. And biomanufacturing improvement starts in development. So when you think biomanufacturing is not your field, it is your field because you have to understand the things in the development. Okay, I think that's an important thing. So it's very close together. So don't think you are not concerned with manufacturing when you are working in the very early start of product or process development. I think that's a very important step. You see here the 3 highlights of Vienna. I can always invite you, come around, visit us in Vienna. But let me go step by step to introduce this problem here. So you see here the life cycle of a bioproduct, biopharmaceutical industry, and you may talk about biomanufacturing here, right? But biomanufacturing is only the end point of the complete life cycle. So the life cycle starts actually here in process development. It goes to our scale up, tech transfer to process validation and manufacturing. Okay, and there are different tasks which we need to fulfill. One is to be quick, accelerated commercialization, very quick time to market. Okay, we take usually 8 to 12 years to commercialize a product, which takes ages, right? We have to be quick. Then, of course, whatever we have developed, we have to have a robust tech transfer. That means we actually have to be successful in scaling up, making a tech transfer and getting to, let's say, manufacturing. Of course, this includes validation steps, especially, right? Of course, finally, we should have no fail batches here in manufacturing, so 0 fail batches here at the end, okay? I think that's the idea, and that's what I try to explain with the bottle, okay? And be sure that this is not only something which runs once, it's a life cycle project. That means we always have to make sure that variations which may come up in manufacturing, we actually have to tackle again in the development, understanding them and make sure that they are properly implemented, validated, and so on. So it's a life cycle approach which we have to follow here. Okay? What is the task? The understanding is actually done by closing this relationship. So you have your quality attributes here of your product, and they have to be fulfilled, that you don't have to drain this bottle away, okay? That means we have to find out how critical process parameters, feed rate, elution profiles, temperature, etc., and raw material or critical raw material attributes actually are linked to the quality attributes, okay? And this one means we have to get to process understanding and we have to control this, okay? So 2 steps, understand and then find a control strategy inside of the understanding, okay? And that's a multi-dimensional problem. You do that probably for 1 step in the production of your unit operation, for example, yeah, here, production of your process, but this is only 1 step, okay? But there may be many, many unit operations, and there may be many of these. So probably you have 200 of those parameters, and you probably have 15 of these quality attributes. That means we have a very multidimensional, very complex problem. When we think about, for example, cultures which we are doing inside of our laboratory, we are cultivating natural killer cells, for example, as really just example cell line. And you see we have many, many issues which we need to tackle. We have to make sure that we reduce the media amounts. We have to know when we add cytokines. We have to probably avoid any other, let's say, additions. And that's a very, very complex problem which we actually have to face. Okay. So how to settle this? So process understanding is also featured by quality by design. Quality by design is something which has come, let's say, 15 years ago, and is the basis for what we are doing here actually to stabilize biomanufacturing. What you need to do is just solving these 3 question marks between quality attributes and process parameters, lots of them. However, this is not easy because they are all related to each other. It's a very multidimensional problem which we have. That means we have to solve the understanding on the product side. We have to solve the understanding on the process side, and then relating product to process here. Okay, so this is really the main problem which we have to solve. This is not a very simple thing, and when you look in literature and also what companies are actually doing, you may know advanced therapies, you may work on also advanced therapies here, and there are lots of OOS, so out of spec events which are happening here with NK cells, with CAR T cells, etc. And companies like Novartis, they have, for example, the license to produce it, but they are not, due to the complexity, which I have just shown before, they don't have the possibility to sell the product because they cannot handle actually the complexity. Okay? So how to actually solve that, and what drives me in this sense? So I'm trying to solve these issues, avoid drug shortages and provide affordable drugs for the full world, and not only for the first world, which only some of us can afford. Okay, and on the other hand, we actually have, as I just mentioned, we have failed batches, we have long time to market, and we have, of course, the drivers of biosimilars and personalized medicine, which we of course also do here. And we have to find a way how to economically optimize biopharm manufacturing, right, actually to get there. And this one, the problem is that we have a big knowledge gap here, a big knowledge gap on the cellular complexity, on the process complexity, on manual interactions, etc. And the solution for that, which we think is really helping in getting this done, is digitalization and data science, to really make sure that we understand our complete system. Okay? Okay. So how to do this? How to get closer to that? So we have first to identify variability. So if we want probably to go one step back, if we want to have stable, robust manufacturing, we have to identify variabilities and actually sort out variability or control variability, right? That means we first have to identify the variability, which we do with model-based data science methods here, also called digital twins. We will come to that in a moment. Then we have, of course, to monitor the variability, and then we have to control variability. All these 3 steps are important. And we have also published on that here with the MIT, for example, with a couple of companies here together, this one. Also, I just launched together here in the ISPE, which you probably know, the International Society of Pharmaceutical Engineering. I launched here the Good Practice Guide on Advanced Therapy, Medicine and Products on autologous cell therapy, where exactly this is happening. And you can imagine that especially for advanced therapies, we have this problem of variability because your blood cells are different from mine. Just imagine this, right? So the variability just comes from the starting material inherently, okay? So there are lots of pain points in cell and gene therapy. I just have listed here some. I will not go too much into detail. Sample number is limited. Sample results come late. Insufficient analytics. All these elements add to this complexity, which I tried to explain before. So what we need to do is probably shown on this very simple sketch here. We have to turn data into knowledge. We have to make sure that we understand the data here. For example, the color, let's say, gives us an information about the maturity or the sweetness of the banana here at that level, but the knowledge is applying the information to a certain objective, right? This is, I think, very important to know. So this is what we have to transfer. We have to get to this level because the objective may change, right? So, and then of course we have also to go into knowledge management, which is an own problem by itself, right? And managing this knowledge because it is a life cycle approach which we are following. So how to do this? Okay, so knowledge is one thing, and then we have to implement this knowledge in the control strategy. So we have to understand where we maneuver, and then we have to make sure we run also inside of this knowledge space. Okay, so our colleague here, Lawrence Yu from the FDA, has published, for example, this pyramid, and we shall get away from the classical traditional approach by end product testing. No, that's only, okay, I have my bottle, I test the quality, and if it's not okay, I put it away. If it's okay, I can use it. That's not what we should do, right? We should go to the level, at least of level 2, where we have actually an understanding, a data-driven understanding, how product attributes and process parameters are actually linked together in a design space, and in this one we can maneuver. However, if you want to really optimize biomanufacturing, we have to go to a different level, with is a real-time automated control level. Okay, we don't have time, especially when we go for continuous biomanufacturing. You can imagine that we don't have time to take a sample, go in the lab, analyze, and go back. The process is running. Okay, so especially for the real-time mode, we have to go a step beyond of this. So we are aiming with our methodologies, which I'm going to show you, to solve or to contribute with methods in the level 2 and the level 1. This is linking me directly to the point how to set up digital twins, okay? Because we may need these digital twins. And what are they? They can be built, let's say, from different data sources. They can be built today. Everyone knows machine learning and deep learning, etc. Okay. However, I want to really be very clear on that I believe that these tools in our industry, where we have very less data and not, let's say, big data like in Google, for example, that we actually should not directly go into machine learning, but we should really go more mechanistically first. So we should start in a very simple complexity of displaying our process understanding, mainly mechanistically. For example, first principles like balances, they don't lie. Okay, this is what we should use first. And then crawl from there to expand to our goal in the complexity, mainly probably mechanistically. Maybe then also data-driven tools like regression, multilinear regression, latent variable methods, or also machine learning, also known formally also tools like neural networks, which we are using. And finally, our digital twin will be here something like a hybrid model. This is the understanding which we did together when I was also doing my sabbatical at the MIT at the Boston in Cambridge. So you see here, I automated that slide. For what reason? In order that you understand. Oh. Sorry, the computer is not doing anything anymore. Just a second. Let me see. No, this one here. So the main Zoom one here. Just a second. Just let's see. Something is. I need to wait. No, it's just frozen. So let's see. Let's give it a moment. Don't you think? You can also log in here with the Zoom if you like, and then we can continue there. You see, I'm playing two computers in the moment. So it's just frozen. Just give me a moment. Probably you can still hear me, but the screen is frozen. Just give me a minute. Yeah. Yeah, no problem. It's not your fault. Yeah. Just give it a minute. Yeah. So we tried to set up Zoom in addition, but probably you can hear me already, so I can talk along that line. So what you should see is here that the digital twin which we are setting up is actually an automated routine, and we are following GMP. This is not good manufacturing practice, what you probably know from a manufacturing point of view, but GMP in terms of good modeling practice. Okay, so we are setting up these models in a very structured format with a proper workflow. That means we start with a goal. We set up these models in a mechanistic or also data-driven way. We calibrate our model very sensitively. We make sure that the model parameters which you are using that they are not collinear. That means actually that you're not having different solutions or multiple solutions for the same problem. Think about an overfit. If you have 100 parameters, you can always model an elephant, right? But that's not the goal, right? It is the art to have only the minimum number of parameters to model what you want to model in the goal, okay? And then you should have only one unique solution and not multiple solutions. That's what we are doing here. Yeah. Can you still count? Can you just try? Oh, okay. So. Yeah, give it a moment. It's done, you think? Yeah. But I have also restarted here, and it looks like I can continue here until you have settled this. Yeah. Just just see. Oh, it hangs up again. Can you share now? Yeah?
Yeah.
Ah, cool. Great. Then we switch. Just a second. Yeah. Hey, there we go. Cool. Just switch here. So you okay? Yeah? Yeah? Okay, so you can hear me still? Is that okay? Yeah? Cool, then we can continue. So the message here is don't start with deep learning or machine learning prior to understanding the problem, right? And the next learning is you should also just follow, let's say, routines which are there, and models and these kind of elements are not so difficult to do. Everyone can do these things. It's not a modeling nerd doing it. So everyone can actually do this today along these kind of these proper workflows which we actually have. Okay, good. The next is that, of course, we are also using artificial neural networks to get this done.
Sorry.
Yeah.
It seems in the participant screen, the screen are still black,
Okay,
but in here it's only.
so let's do new share. What about now?
Yeah.
Hey, there we go. Okay, no worry about that. Super. Okay, so use those kind of routines. They are there. I don't want to go too much into math here. Just the message is, this is possible. Oh, no. You always find people which know math, and that is simple to do. It's a routine which you can do. We don't have to reinvent it. Okay. You always have these publications in here, and you always can ask me to send that. You can find these publications along that line. So you also have possibility to use, of course, artificial neural networks to help you properly in this way. So these are machine learning tools which are integrated. When you actually look at this slide up here, so we actually have possibilities to link machine learning parts with mechanistic models, etc. I also don't want to go too much into detail, but those kind of hybrid models can be done just to be there, okay? And then you actually get wonderful good fits of your cell growth kinetics. For example, you can identify, let's say, proper rates. You can identify proper reaction scheme of your cells. But more importantly for me, let's say, of this lecture is not to show you how to develop a twin, but more how to deploy and what are the benefits of that approach. Okay, that's more important for me. For example, if you have this digital twin, what can you do with it? For example, offline. For example, now you just have this model, what to do? For example, for experimental design, what can we do here? So we are not doing trial and error. We are not doing classical design of experimental tools, probably as you know them, a multivariate approach in different directions. But we are going to model-based experimental design. Yeah. So these approaches, like elements on statistical design of experiments, are explorative approaches. That means actually you don't know anything and you shoot in all directions, if you see what I mean. Okay. But if you know something, like you have a model, use it for experimental design. Okay. You will be much quicker. You will need less experiments. You will be much quicker in finding the goal. And let me explain this with a little example where we have here a cell culture run of CHO cells, Chinese hamster ovary cells, for example, for producing monoclonal antibodies. So normally you feed at these moments, you feed every day, you feed a kind of glucose, a bolus shot every day, okay? and normally you feed here the red amount of this, right? And then you reach this kind of concentration of the limiting component, which was, let's say, the certain amino acid in the medium, okay? And with the model, we actually said, what would be the best feeding strategy? What should we add, when, how much we should add in order to optimize productivity? That means, for example, going from this productivity, so that's a standardized titer, going from this productivity formally to higher productivities. And here you see that the model, if we use this for an objective optimizing function, that we can actually achieve more product if we feed not in the red way, but if we feed in the blue way, yeah, resulting in this kind of strategy. So you see, if you have a model, you can optimize very quickly and be much more directed to optimization of your process. That's one thing. You can use the digital twin in offline mode for experimental design. If we now move this approach into a real-time mode, okay, then we can, of course, do a little bit more. We can probably really control and optimize the system. Let me show you the general approach which we are following in our, let's say, research along the complete life cycle of the product. And then I come to the digital twin real-time implementation. So here you see again the task, okay? The relation of quality and performance to process parameters, raw material attributes. That's the process understanding. We have to make sure how these two things are related, right? What we are doing is first analyzing the quality and the process performance. We are doing an inverse analysis. We are trying to solve this by proper measurements, process analytic technology, by statistical tools like data cleansing, outlier detection, filtering, all these kind of things. We get to an understanding relating the inputs to the outputs in this multivariate complex way, like what we have seen before. And once we have this understanding, we put this into the digital twins, and I've shown you how to build up these digital twins, okay? And in order to really make use of these digital twins, we actually implement them in real time. And then we can do optimized or predictive control. And this part of the lecture here, I will mainly focus on this part now, okay? How we actually can really optimize this process. This is shown here at a process which is done, for example, protein refolding here. It doesn't really matter. It's just any reaction in biology. Here, this is refolding the process from recombinant proteins of E. coli, for example. What did we do here? See here actually what we are doing. We have here the reactor. So here we have the physical system on this side. We are measuring, let's say, the different reaction entities like native protein, solubilized protein, aggregates, etc. We get these data into the digital twin, and then we deploy this in real time. That means we can deploy that in a state observer or in a model predictive control. And I will go both ways with you. Here, we first have a possibility to predict what is going to happen. It will be a process event prediction system, okay? So we run that and we can see what is happening. And then, of course, if you close this loop, you can, of course, also control back what is happening. I will explain you that in a moment also. So here, for example, you see how, let's say, the measurement points of the physical system are aligned with the digital twin, which is running in real time, and there is a certain estimator which is actually doing this properly, right? So it's estimating and actually matching these two physical and virtual systems to each other. And then you can actually see a prediction what is going to happen in your culture. So where will it go in the future, okay? So that's event prediction, very helpful. And with this one, we actually can really increase here, for example, the yield in manufacturing almost double. So we can increase to double amounts, really high yields, okay? So we are not, as you see, we are just not doing, let's say, in silico work on the computer. We do real lab work. So if you would come around to us, you see this kind of lab. I just explain you how that looks like. We have here, for example, the bioreactors here, which we have spectroscopy devices for directly measuring inline metabolite, substrate, product qualities, etc. We can take a sample of the reactor, we can filter this, we can make retention samples out of the reactors automatically. So we have every 6 hours or 2 hours, we have a sample. And then we can inject these things automatically here in HPLC systems, in photometric analyzers, and so on. So it's an integrated tool which we actually have here, okay? And like this, of course, you see the physical system, and the virtual system, the digital twin, is of course on top of this, yeah, which you of course can't see on this picture, right? In this. So what we actually have seen here first was you have your process, you monitor your process, you look at the digital twin, you align, let's say, your digital twin with this, and like this, you actually get to this prediction, what I have just shown, okay? So you link the twin to the process in such a way, and so on. So that's only the observer part, right? So you are actually only seeing what is going to happen next step. But what is probably even more appealing is if we now close the loop. That means we're actually going back, okay? And we really control on an objective function. And therefore, I would like to get you a little case study, which we have done here, which is here showing a classical problem in recombinant protein production for, let's say, medicinal products. We have here the specific productivity over time. That means the cell is producing normally very much of this product at the beginning, and then it actually drops down steeply, right? The better way would be to have a more constant production, and you can imagine that the integral under this curve is higher than this here, and it enables actually continuous biomanufacturing if we would go there. Even if you are not going into continuous biomanufacturing, you can imagine that whenever you harvest, if you harvest here or an hour later or here and so on, that your load to the purification of your protein will be completely different, because you have lots of lyse cells, host cell protein impurities, and so on, which really gives a load to the downstream. Yeah. So in any case, it's much better to have a more constant production over time, okay, and not that dynamic. So what is normally done in this case, you see here, for example, you can feed your inducer or your energy substrate to the cell in different levels. And you already see that it makes a difference. If you feed high, you feed like this. If you feed like this, you actually get that curve. So it looks like it's already better. But with the digital twin, with the digitalization solution, which I show you here, we can do another thing. We can go back. That means we want to make sure that this is constant, and we need to derive how should we feed in order to get there. You see the control problem? Yeah. So that's what we try to solve. And if you have a digital twin in a real-time mode, then you can do this like this. Okay. This is the way how to stabilize biomanufacturing here, and of course to increase also productivity. The next slides are very much mathematic, and I will quickly jump over that. I don't want to lose you too much. But what we need, of course, is a model. We need a digital twin, whatever that is, but we have a model. I think that's one thing, definitely. Secondly, we have to characterize that model for, let's say, different feed rates, and we have to validate this model that actually the the estimation is okay, right? These are all good practices in order to develop and characterize a model according to good modeling practice. And then we actually have to know how do we want to feed. So you see, normally this specific productivity is doing this kind of thing, and now we use our optimized experimental design approach to see how can we maximize productivity, so that's the bold curve, and how do we need to feed to get there. Okay, so how do we need to feed? So that you see up here, so that's the feed. How do we need to feed in order to get to the maximum productivity? Yeah, I admit it's still not constant, as we actually saw here. It's not constant, but optimized. Okay, it means how do we need to feed? We need to feed something like this. You would never thought of this, right? It would have been never thought that this is the right way to do. Okay, so you also see that the product quantity, of course, it's high, and it's over a long time very high. I don't want to go too much into detail how we solve this. Here we put our model in here. It's a highly non-linear problem, if you can imagine. Yeah, it's highly non-linear. However, we have in our system a linear feed. We finally want to establish a feed, and therefore we need to do some math. And I like this picture because it shows a little bit what it is about. So it's probably a simple equation first, and then it's getting quite complicated. And those kind of elements are the let's say, for example, Lie derivatives, which we can calculate from here. I will keep it simple. I can just mention that master students in our lab are doing this. Okay, so we can master that. But our team is very much constituted from many, many different persons. We have biotechnologists, we have geneticists, we have statisticians, data science, process engineers, electric engineers, and so on. So all of that together, and then we can achieve that. But this is, it looks like rocket science, but it's not. Okay, we just took this kind of non-linear feedback linearization tool here, actually from a different market segment. It was from automotive industry, and we put it into biotechnology. We didn't invent the algorithm. We just transfer algorithms to this new field. Okay, and I would like to leave you with this. So what we finally get out, we get to the control of the best feed rates, etc. And we also get finally to very high productivities, much higher than what you would have done with classical constant feeds. So this kind of, let's say, very strange feed rate, which we had, really led to higher productivities at the end. Okay, so these are the potentials when we link, let's say, our understanding via digital twins into the process back. Okay, that helps to really optimize and also stabilize the process. Okay, so here is a little workflow which I want to give you for this. So we need a model. Yes, we need a model. We have to calibrate and check the model. We have to use it for optimum experimental design. So how should the feed rate be? We derive a control law, so this kind of math thing, what you have seen, and then we can deploy it and earn our fruits, right? And then, of course, finally, we have to make sure that whatever it does is also, let's say, scientifically sound, okay? So you can also find that in this recent publication of 2022, which we are very, very proud of. Another example I want to show you is the real model predictive control. So the controller not only looks back what has happened and what is, let's say, to be done in this moment, but what is going to happen in the future and how can I optimize performance in the future. Okay, so here you see a comparison between a classical, normal controller, which really follows the line, okay, which follows the normal behavior, and you see the behavior of a model predictive controller, which looks forward, and you see actually here that it deviated from the set point. And the question is, why does it deviate from this set point, right? And it did this because it has seen if I would run in this way, I would produce a metabolite which I don't want to have produced and which would take productivity off. Okay, so here you see productivity was sustained, it was higher in this red case rather than running the PID control. Yeah. So here these tools are really helping predicting process events and even compensating for that and making sure that these events are not happening. And then we increase productivity. This example has been implemented with a very large scale in manufacturing for Novartis in Austria. Yeah. So this is for industrial biotechnology, not really for the biotech, biopharmaceutical part, but for industrial biotechnologies, we have implemented that and also published along that line. So the next step, the last bigger chapter of my lecture is not only looking at one unit operation and understanding this, but looking at the complete process chain. That means not only optimizing one step, but finally what counts, think about the bottle, what I started with, that we actually have to make sure that the final product quality here at the end is okay, right? And this is a problem of 200 parameters, which of course are interlinked. It may be that even for one step, you are running that inside of the boundaries and you are happy how you run it. But you run it a little bit off the set point, a little bit off, but still inside of the region which you qualify. The next step, you run a little bit on the left side of this process parameter. And you can imagine that the interactivity between the parameters finally will probably lead to failure, right? So if you change the parameters and the interactivity between the parameters finally leads to the failure. I have seen that many times. And if you have this bottle problem, it doesn't come that some operator has really run its process in a bad way. I don't want to say that, but it's the activity. Some people have operated a little bit on that side, the other one have operated on that side, and the propagation of this error is actually failing then finally in the product quality. So we have to avoid that. So what we have to analyze is what is the sensitivity of any process step here on my drug substance, my drug product, or even directly on the patient. So we are now looking also at the patient side, right? We have to understand what is the probability of failure if I would deviate a little bit upstream here, okay? What would be the issue here? And this is how we actually need to solve this problem. We cannot look only at the manufacturing level. So that's what I said with the introduction. It's not that we only look at the manufacturing and we run 20, 30 batches always in the same way to get the understanding. Good manufacturing starts with understanding the points in process development, in process characterization, right? And we have to make sure that the data from here, so you see that's an iceberg, so the tip of the iceberg here, right? But the real understanding is happening here, yeah? And we have to include all of that data in understanding the manufacturing robustness, okay? So that's what we are doing here. So we have put this into a software, and you see here that for some unit operation, you have a certain variability of your input parameter and a certain output of the input of the output. Okay, you have a certain output variability, and this output is of course the input of the next step. The output of one step is the input of the next step, and this is propagating over all these steps. And now you can do, for example, Monte Carlo simulations that you find out what is the probability of failure at your drug substance or drug product level. Okay, so this is what we are doing. For example, you can change the output quality, for example, of that step. you change the mean of the output of this step, and you understand the probability of failure of your certain quality attribute here. Or inversely, you find out what is the accepted quality at that level, right? In order to make sure that your product quality here is okay. So that's one thing. And then, for example, you can find out what are the real boundaries. So you see here a plot for a certain quality attribute. You see here the different unit operations, and you see how this quality attribute is actually run over the different unit operations. Each of them are observations, and you see what kind of boundaries I really need to set. And the integrated process model, which I have shown you, is actually giving you this lower boundary as the acceptance criteria. And you see here it is more stiff than a classical 3 standard deviation approach, right? Which are here the orange lines. But here it is relaxing this, okay? So it gives you a proper probability of failure. Inversely, you can set your control strategy also very nicely, like changing here, for example, the process parameter, like an elution pH of a chromatography. If you are doing that, so you are changing here your elution pH, and then you see what is the probability of failure at the drug substance level. And inversely, you can say, what is the upper proven acceptable range for that step? Of course, taking into account all the understanding along the complete process chain. Yeah. This is what digital twins are for. We are really making predictions of the success of our process. And of course, all the development data are part of it. We also can move these things in the real-time mode. That means actually we are probably running 3 steps already in real time. So the process is running and we know how this process has run, okay? And then you see here the probabilities of our process parameters are almost none, because the uncertainties, because I know exactly how they have run. And then I can predict, if I would run with these process parameters, what will be the product quality finally at the end, and what will be the probability of failure at the end of the process. Yeah, those kind of things we are trying to do with these kind of tools. This was the bright side of the moon. That means actually, these are all the potentials which we can do with digital twins. However, what we need to do some homework. Yeah, and these are the prerequisites which I would like to show you very quickly. We have to make sure that all our data are properly connected. That means we have here the unit operations of my process in one dimension. We have here the life cycle of my product in the second dimension. And in both of these elements, we have lots of different data sources. We have time series, we have quality data, we have spectra, we have images, whatever you can imagine, right? Cellular attributes and so on. And they may come from the development in the earlier stage of the process. They may come from the manufacturing at the later stage of the process. And we have to link all these data sources together. We have to put them all together in one data model. We have to make sure that we can contextualize the data. That means we actually want to find out, okay, how is the relationship of this phase of the process, right? How is this related in this step to the quality attribute at that step? So we want to have any relation. Yeah. So data contextualization is very important. If you would just dump all this data into a big bucket and you shuffle this and you try to run multivariate data analysis over this, it will never work. Garbage in, garbage out. Okay? So you really have to get from the domain, and you are domain experts. Look at your data, cluster your data, set phases, analyze what is the variability in your data, contextualize the data sources, and then we can really get to the proper information. So what we are featuring in this way, so and you have seen my double affiliation. So I'm a full professor on the one hand, but I'm also advisor for Körber Pharma. And the point is that 10 years ago, I opened a company which was focused on data science and data management in software and consulting, featuring validation and so on. And this company, so my startup company, has been bought 2 years ago from Körber Pharma. Now we can do businesses in this way and deploy the software for your purposes in the same way. Okay, so we are using that in development. We are using this software in manufacturing. We are using this for early, let's say, process steps, for late process steps, no matter. Okay, so we are feeding the data all together. It's a software as a service tool. It's something which you can just run directly on your computer. It doesn't need any installations, etc. But let us see what the software is doing. So it actually links all kind of different data sources together, but data collection is not enough. Only data collection will not help. I hope I could show you that data contextualization is a very important step. We have to bring all the different data sources in relation to each other, that we can actually do proper analysis. So we have to put this into a unique data model, and then we can start doing our work, what we are trained for, what we were educated for, right? We are not educated for data management and copy paste of Excel sheets. We are educated for using our brain, right? And therefore we facilitate the former parts of that by domain-based software, and then we have a direct link to analysis, calculation, reporting, visualization of the data. I will not go too much into detail. I want to stay scientific here, but if you like to know more about that one, we can, of course, jump in later. And of course, important is that you're not only doing your work, but that you're also sharing your results with different partners. You're not alone in this world. You're talking to so many different groups inside and outside of your organization. So be ready to share your data, share your results, and so on. And I believe that all of this needs to be together. So we have to have seamless data management and data analytics. So closing my talk here, I started with this pyramid here. And this pyramid actually projects to different digitalization solutions, which are here linked in this data maturity model. So it's not about just computerization and connectivity, okay? Just connecting the data. It's about seeing what is happening, okay? Visibility of your data. The next is understanding. I think I talked a lot about understanding of the data, right? It's very important. But then be prepared to what will happen. So that's a predictive part of it, okay? Know what is going to happen. I hope I could show you this one with a model predictive controller and these elements, that I know my product will be okay, that I know how I need to run in order to make sure my product quality is okay. And then last but not least, it is not only done once, it's a life cycle approach. We have to adapt. We have to adapt our process, and we have to adapt our understanding and our digital twin in the same way. So also the digital twin has a life cycle approach, okay? So we need to cover all these levels of data maturity, okay? I think organizations are in the moment mainly in level 2 to say, okay, what is happening, and probably understanding, but we have to look in front to see, we have to be prepared for what is going to come. Conclusion in that, we have independent models which you can develop. I have shown you a couple of them. We can integrate them into the process chain. So whatever knowledge you have available for your process, they can be integrated into the overall holistic analysis of the process chain. This one can, of course, lead to what I have already shown you, to setting the control strategy very properly. I have shown you this example. And this one is the way to set the control strategy properly and to make sure the tech transfer is quick and successful. And then, of course, the digital twins can help in the product life cycle here to predict what is going to happen, also statistical process control, and also here showing process events. All these things come directly from the software. And with this one, we actually cover lots of different business cases. Here, the control strategy. I showed probably here deviation management. Also, real-time batch release is a very big issue. You can actually release your batch with a prediction, what I have shown with a digital twin. Lots of business cases can be addressed when you employ these kind of tools. I published a couple of books here. Probably they're interesting here for you as well. There are digital twins. There are probably 15 chapters in there and about 20 definitions of a digital twin in there. No matter, it depends what you want to do with this. What is your goal in this direction? And I hope I could show you that we actually can address with the digital twins optimum and targeted experimental design. Think about what we can do in offline mode. We can predict process events. We can make closed loop process control with that, really to optimize it. It is also a lifecycle companion for continuous improvement. And I hope I could show you with my lecture that all of these elements are very beneficial, and they address business needs, they address biomanufacturing, but they start all in process or product development. They really start very early. Never leave this stuff to manufacturing guys only. Okay. So you see here, we have different steps to really get to proper control. We need good data quality, data integrity. We need a holistic data management. I hope I could show you that it's important to fuse development and manufacturing data. We have to include your domain knowledge. We have to include you and your domain expertise in setting up the control strategy and include that in the data sciences which we use. And last but not least, of course, the digital twins are the tool to capture the knowledge which you have and deploy the knowledge, right? And that's the way how to set up these control strategies. With this one, I finish my talk and thank you very much for your attention. Thank you.
Thank you very much, Prof. Christoph, for your amazing talk. So is there any question from the audience? Well, while waiting for the audience to type their questions, I'm going to ask question for Prof. Christoph. First, why digital twins? Why the name is digital twins? Is it because is it real-time monitoring?
Let's say before we have set a model, okay? So we have a physical world and we have a virtual or digital world, okay? So first we have probably 20 years ago, we set a model. Now we call it a digital twin. It's a little bit close to this. So we say the digital twin is the real-time version of the model, right? If we really get to the real-time deployment and implementation of the model, then we get to the twin, okay? Yeah?
Does the solution also predict the saturation of the system?
Yes, of course. It's always about prediction. So as you have seen, we can control, but control is a very long way to get to closed loop control. The easier way is the prediction part, and only, let's say, you can use the digital twin for getting trust. Okay, so you want to probably say, okay, if I see the process, I think it runs like this, and the digital twin tells you probably, yeah, it's really true. So you get trust in this digital solution. Okay, and without trust and without taking the society in its acceptance, we will never incorporate that in our process. Okay, so the first step is always, do you have trust in the system? Does the recommendation or the prediction is okay from what I think? And once I have the trust, then I would go the next step. Okay, and then probably implement that later on in a closed loop. But first, it's about prediction. Yeah.
And then does this digitalized solution also can predict any product biomedical process manufacturing?
Of course, we are doing this, right? So, of course, I'm talking mainly about the processing part rather than the product development part, right? So, so normally you have product development and then you have process development, right? And I'm more in the process development part once the product has been defined, okay? So these solutions which I show here are solutions for getting the right quality of the product by controlling the process properly. Okay, I think that's what we are focusing. You can use other digital twins for product development also. Yeah, when you want to know, okay, how do I need to append or modify my product in order to get to a certain function, then you would use different digital twins. Yeah.
So in the whole process of the bioprocess manufacturing, what are the key factors in deciding the best technique for certain process? Because there are so many elements there, and then what are the key factors that decide say the best technique for this kind of bioprocess?
Yeah. It's a very big question. The key factors are what you know about your product. So you have to understand what are the influencing factors on product quality. So on your bottle, okay, for example. Yeah. So and if you know that your product quality is rather okay, whatever you run, the process can also be simple. But if you know your product quality is very much sensitive to certain parameters, you have to dig deeper and to control this properly. Yeah? So there may be products which are very simple, and there are products which are very complicated. And accordingly, you have to set the complexity of your model or your digital twin in the same way. But it all goes via the process understanding work, yeah, which you first have to do, right? And then you go from there. And this one starts, I tell you it again, very early in process development. You have to understand that, right? And if you only start controlling your manufacturing process without understanding, it will fail, right? I can just show you a classical thing here. This kind of statistical process control. So you have a couple of runs, and you say, okay, they always run, and you put a certain band over this, 3 standard deviations, and then you should always be in there. However, if it deviates, if the new process deviates at a certain point, it's not anymore in this yellow field. What are you doing? In manufacturing, then normally people come together and say, oh, now I have a deviation. What shall I do? And they say, let's try this. But this is not what we should do. We should know why this deviation is happening, and we should know how to properly compensate for this, right? And this is still not there. Yeah.
So there's one question to the audience.
What are the biggest challenges in implementing? There is the question in the chat box. What are the biggest challenges in implementing digital twins in biomanufacturing? And which countries use digital twins the most for biomanufacturing of regenerative medicine? And what should we do as biomedical students to understand about these digital twins?
Let's say step by step, right? Exactly.
Yeah.
So the first question is, what are the main showstoppers to get there, right? This battery is running low. Battery. The showstopper is not the IT architecture. Okay, this is available. The showstopper is not the way how to set up these digital twins, because these workflows are there. The showstopper is to relate all the data sources properly together. Yeah. So what you should really look at. So I take that again. I take that picture here again.
Yes.
How do you do also the biomedical students you have? Okay, how do you relate? Currently, you have some measurements which you take at the beginning. You take some measurements at the end. You may have some time series. You have some pictures, some spectra. Yeah? So how do you currently link these data sources together? And the showstopper is that we have not understood how to really contextualize the data, right? You have a picture. What do you do with the picture? You look only at the picture, but you should extract information from that and link this information to other data, which can come from a different time point, or right? And I think we have to understand how to deal with the data. That's the main showstopper, in my opinion. Because data we have enough, but data analysis we are not doing. So the showstopper is to bring data analysis up to speed compared to the data generation, which is much more than the data analysis, right? That's a showstopper. Could I answer your question in this direction?
Yes, thank you. And then another one, which countries use digital twins for the most for biomanufacturing of regenerative medicine?
Yeah, so this is really when I'm frequently in the US. We are doing lots in the US with this. I've been 2 weeks ago. I've been in Boston area, so East Coast in the US. Big companies are doing this. Takeda is doing that one. Biogen is doing that. Amgen is doing that. So the big companies. In Europe, we also have companies like Merck, like Sanofi, and so on. So they're using these things, and they know that this is required. And for example, this software tool which you have seen here, which is deployed for worldwide visualization of development data inside of Sanofi. They have 1,500 licenses worldwide to get all the data together, and then you really make a difference. Yeah.
Okay. Thank you. Then maybe the last question is: we know that manufacturers have strict standards in the processes they will implement in producing a product, and of course carry out various validation on various parameters. What are our concerns about the quality of existing products, so that we look back to make sure a product provides good benefits for us for use?
Yeah. There are two levels. There are new products. Yeah, and then of course this approach has to go in the control strategy, and you need to put your BLA, so the biological license application, you have to include this understanding, and then it will be fine. It is a different way of thinking when you actually do this. For existing products, we have to go step by step. We have, of course, a license, and we have already filed this. And we have to make sure that we go step by step. We have a certain deviation, for example, in an existing product. And then the question is, how do we want to solve that? Do we want to solve only this out of spec right now? Or do we want to solve this deviation with a holistic understanding approach? Okay, so I see in many cases that now big companies are starting, even in running processes in existing products, to actually, for certain unit operations, to bring in holistic process understanding. Okay, it's possible for both ways. Okay, the full-blown approach for new products is of course clear, but it can also be applied for existing products. The people are doing this. Yeah.
All right.
Yeah.
Thank you so much. Is there any other question from the audience? No?
Thank you. Thank you for your presentation, Professor. My question is, in regenerative medicine, biomanufacturing of these products in regenerative medicines, they also use a decentralized approach. In contrast with the centralized in a cell processing center, but they use also there's equipment that can be used in clinical settings.
What's your question?
Yeah. So these equipments used in clinical settings also release products.
Yeah, just asking this. Yeah, of course, it is for decentralized solutions in the same way. And you have variabilities which can come from the patient, decentralized. You can have variabilities which come from different devices which you use in a decentralized way. And you have to understand the root of this the root cause for these variations. And you have to build the control strategy, including those kind of variabilities. Yeah. But that's definitely possible for autologous cell therapy. Absolutely, for example, right? That's why I just want to mention that guide here again. Yeah. Good. Any further questions? So, yeah. Yeah, absolutely. Yeah, I fully agree that when I look at that picture here again, so for the audience virtually, the question is how to deal with the input variability, which may come, let's say, from the donor or from the primary cells, etc. Right? And that's the reason why I get big to this picture. So in, let's say, classical, let's say, recombinant protein production, you probably have more variabilities on the process parameters on the upper side of that picture, okay? But in autologous cell therapy, you have it much more on the left side, these critical material attributes which get in there. And this is not only the material of a buffer or a serum, it's also including the cellular attributes of the primary cells, okay? So which is inherent. So the variability of the starting material is inherent to this problem, right? And there are two ways which we normally do in solving it. The input can be or needs to be very properly monitored. So we characterize material attributes like cellular components via flow cytometry or different other analytical techniques, yeah, to really understand the cellular behavior or the cellular attributes. You analyze pictures, you make automated image analysis, okay? So you are really analyzing this part here, you're really analyzing that with holistic analytics like flow cytometry, image analysis, also lots of different attributes to characterize these cells. And then you link that in. And the other way what we are doing, the second part, is modeling input uncertainty. That means we know that the variability is, let's say, is there, and we don't neglect it if we close our eyes, but we are aware of that, and we model input uncertainty. So there are possibilities to include variability in the input vector and then still have a proper process. So these are filters which we are using. So these are filterings on the input material, and then you can get still to a process. But it's actually, if you Google that, it's under modeling input uncertainty, okay? These two ways we have, and mainly we do both. We do the analytics and we do the modeling of input uncertainty in order to get this this problem really solved. And this is a real one in autologous cell therapy.
So if that's the case, then in order to generate data for your models to work, definitely you need lots of trials. Cells, and I guess you should actually set some standard. When you do this analysis, then you find out that it doesn't meet the criteria that's predicted good output. From the beginning, you just discard this kind of cells. Otherwise, because with primary cells, like you said, lots of inherited attributes that very difficult to control. For instance, you have this elderly man, and then flow data showing some thirst in the surface.
Yeah.
Good, it's not sufficient. You probably can actually say it's not recommended.
So it comes back to, you need a very proper experimental design. There's no doubt on this. That's the reason why I go to this picture here again. You need very good experimental design. You have to understand what are the variations which you have, okay, which could be there. Of course, we start also in cell therapies. We always start with statistical design. Yeah, we probably don't know anything. Right? Then we have to properly disturb the system. We have to properly shoot in all directions and to see what is happening. And then narrow down what are the elements which are really important for my product quality. Okay? And it's not the number of experiments we really need to fix, but it's really the meaningful planning which drives the number of experiments. Think about if you have 100 runs in manufacturing, which supposedly you all run in the same way, right? Yeah, we are dynamic. We are problem solvers. Right? So when you think about the real quality by design approach, it is never. How do you do that? There's always a risk assessment. You think about an Ishikawa diagram, for example, a fishbone diagram. What parameters could influence my quality? And there may be 100 of them. Okay, and then you start saying, come on, what do I know about this variation, about the influence? And if you don't know anything, you boil it down to an experimental design, like what I showed. Once you have done this experiment, you go back to the risk assessment, okay? And you see if now your risk assessment is better. And then you reassess that. So proper development here is always a link between risk assessment and meaningful experimental design, okay? And it's an iteration. And then you get to it, okay? But don't rely on manufacturing data only. Think about the iceberg picture, okay?
And last question, how useful is it to help us from the regulatory scale to maybe
if, let's say, the digital twin has to have a goal, okay? If the goal of the digital twin is to characterize scalability, then it is useful for this. So probably a little bit strange answer. But first you decide what the goal of the digital twin is, okay? In this case, here, I have to go to this. So in this case, the digital twin was only built for estimating tighter. But probably you build a twin to understand the sensitivity of scalability of your system. Okay, so how sensitive it is of your cells to inhomogeneous mixing or addition, ingredients, etc. Then you build a different model for this. Okay, and stage one validation. When you think about, I don't know if you're aware of stage one validation, which is process characterization work. Okay, there. An inherent, let's say, requirement is a scale down model qualification. Okay, so it's not about scale up, but it's about scale down. What is the difference? Scale up is exactly so you try to make that correct what you have done in small scale. You try to get it in bigger scale. Scale down is a different thing. Scale down is understanding the effects of your large scale properly in a small system, you see? So accepting the variations in large scale, but understanding them. And then making sure in the small scale to understand what is happening and even predicting what is happening. And therefore, for this digital, for this scale down model, the digital twin is actually the enabler, okay? So it really helps you understanding what is happening in the large scale, and then you really can model this in small scale, predict in small scale, and have very less experiments to really verify that you understood the large scale thing. Yeah, but it depends always on the goal or on the model, if it can help you for scale up or tech transfer. Yeah. So it's never the case that you say, I have my data set, I want a model. Tell the goal. So we need more time to fix the goal than developing the model for this goal. That's interesting. Yeah.
No further questions. Thank you very much, Prof. Herwig, and the audience for very active participation. This has been a very dynamic webinar in which 3 laptops are used by the speakers. And we have learned many different aspects of biomedical field today, which is improving biomanufacture of regenerative medicine using digitalization solutions. Prof. Herwig showed us that we can use data from the process in real time to get the knowledge needed, such as how much to add, when to add, and what to add to optimize the production. And with the global population overburdened with many communicable and non-communicable diseases and medical costs, this could be one of the solutions to provide affordable medicine for population. Right. So, Prof. Herwig, is there anything else that you want to say to the audience before we close this webinar?
I'm very happy to be here.
Mhm. Thank you very much, Prof.
I'm just very happy to be here. Thanks for that. I know this is not exactly the field you're working in,
Mhm.
but I think it was important for me that you see that this one will be the future, even in your fields.
Mhm.
And the potential is not always just found in lab work. The potential in your work is done by analyzing your data you have or you will have. And I think that's very important. This one will increase efficiency and, of course, finally improve patient safety, right?
Yes.
And affordable drugs, right? This is for me very important. I run after affordable drugs, affordable, right?
Mhm.
And I hope I could contribute a little bit that you think on probably doing your work in a different way and are open for these digitalization approaches. Thank you.
Wonderful. So, Prof. Herwig, if the audience wants to contact you, where they can reach you for further questions or other things?
Yes, of course. You Google me anytime, right? You will find me. I'm a very public person, but I think my contact data are also just here at the end. I just show them again. So just contact me anytime, reach out to me, come to Vienna. Very happy to be here and to have met.
Awesome. Thank you very much, Prof. Herwig. And with that, we close this webinar. Thank you very much, everyone, for attending. Have a good day, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Ladies and gentlemen, we finally come to the end of today's agenda. Let us close the webinar by reciting Hamdalah. Alhamdulillah. Those are the agendas we have presented to you all this morning. We hope you learned something and thank you so much for your attention. See you on the next occasion. Wassalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh. Thank you.
Izin live ya.
Izin live. Terima kasih banyak. Thank you so much.
Baik, terima kasih.